Hello, this video is about specializations in the erythrocyte, which allow it to do its job of gas exchange. There are three things that are unique about the erythrocyte. One is that the erythrocyte has no nucleus. We saw that in the development of the erythrocyte when just before the reticulocyte stage, it gave up its nucleus and really all of the other organelles, including mitochondria. So the red blood cell also does not have mitochondria. And if you recall, mitochondria are important for synthesizing ATP for the cell, which means that the red blood cell is going to have another mechanism for making ATP. Finally, every red blood cell has lots of hemoglobin, the protein that actually binds oxygen and helps to carry carbon dioxide, which we'll talk about when we get to the respiratory system. Now, getting rid of the nucleus has a disadvantage and an advantage. And whenever you see this in biology, the advantage outweighs the disadvantage, or else the change wouldn't take place. So the reason that the red blood cell gets rid of its nucleus is because this gives it an increased surface area to volume ratio. Now remember that surface area is what determines how quickly diffusion can take place. The greater the surface area, the greater the rate of diffusion. So anything that increases the surface area relative to the volume of the red blood cell is going to make it easier and faster for oxygen and carbon dioxide to be exchanged. For those, I said ration. There we go, ratio. Now, to make that make a little more sense, let me just give you a scenario where I go and I buy 2 balls of pizza dough at the store and I'm going to bake them at home. And one of them I leave as a ball like this, but the other I flatten out so that it looks like a pizza. Now, which one of these is going to bake faster, right? The answer is the one that is shaped like a pizza, because it is flat and therefore the heat can penetrate deep into, or deep enough into the pizza to cook the dough. When the pizza dough is in a ball, the heat can only penetrate so much in a certain amount of time, and so the outside will cook, but the inside will still be raw. So you can kind of think of red blood cells the same way. Red blood cells, because they've lost their nucleus, which would make them spherical, they are now flat like the pizza dough, and that means that oxygen and carbon dioxide can move freely into and out of the cell and do so very quickly. All right, so the reason for no nucleus is to increase the surface area, or the surface area to volume ratio. The disadvantage of having no nucleus is that, let's call this disadvantage. The disadvantage of having no nucleus is that we can't make new proteins. Right. The red blood cell does pretty fine with this, except that there are 2 proteins. One is called spectrin, and the other is called actin. And these proteins sit on the inner surface of the plasma membrane of the red blood cell, and they help to make the plasma membrane more fluid or bendable. This is important because red blood cells, once every minute, pretty much, end up having to squeeze through really tiny capillaries. And the capillaries are so tiny that the red blood cells have to fold and squeeze and change shape in order to get through. So the spectrin and actin are important for that. And when they run out, because we have no nucleus, we cannot make more spectrin and actin. And for that reason, red blood cells die at the end of 120 days when they try to bend one last time and they can't do it and the membrane ruptures. All right. The next thing about red blood cells is that they lack mitochondria. Okay, they lack most of the organelles. They choose instead to fill with hemoglobin, which allows them to carry on the process of gas exchange, highly efficient gas exchange. But one of the organelles that's missing is mitochondria. Now, if you recall, mitochondria are the powerhouses of the cell, and this is where we use oxygen in order to convert glucose breakdown products into ATP. Now, if you think about it for more than a second, you'll realize that it doesn't make sense to have mitochondria in the red blood cells, because if mitochondria use oxygen and red blood cells are trying to transport oxygen to the body, then there's a conflict of interest. So by getting rid of the mitochondria, red blood cells are able to this prevents them from using the oxygen they carry. That's the advantage. The disadvantage is that they can't use mitochondria to make ATP. And so the disadvantage is that red blood cells must use anaerobic fermentation in order to synthesize ATP. And anaerobic fermentation, if you recall from the first semester of A&P, always results in the production of lactic acid, which, when that builds up, becomes toxic to cells and tissues. So this presents us with a situation where we have to deal with the consequences of having no mitochondria, but having no mitochondria is essential to the function of the red blood cell. Finally, red blood cells are packed with hemoglobin. So, as I said, about a third of the red blood cell is packed with hemoglobin protein. Now, hemoglobin protein, there is a slide that shows you a pretty picture. I will draw my own version in just a moment. But each red blood cell has approximately 280 million hemoglobin molecules or hemoglobin proteins inside. So 280 million molecules of hemoglobin per cell. Now, each hemoglobin can bind 4 oxygen molecules. So each red blood cell, if it has 280 million hemoglobin proteins, and each one can bind 4 oxygen molecules, that means that each red blood cell can carry more than 1 billion molecules of oxygen, which is pretty impressive for a tiny little cell. Now, what does the hemoglobin look like? It has 2 parts. It has a protein part known as the globin. And it's actually 4 different protein chains. So there's one protein and another. And one and another. And these are held together in tertiary structure by disulfide bonds. And then inside each of the globins, there is a heme group, which is a heme pigment. In fact, it is what's responsible for making the red blood cell red. Now, each one of these heme groups binds 1 molecule of oxygen. Making it 4 molecules of oxygen per hemoglobin molecule.